Hi everyone, welcome to Fun Biochem Workshop Series number 19. Today we're going to cover bioinformatics, and I'm Emily Wren. So an overview of what we're going to cover today. So the first part is an introduction to bioinformatics, then the central dogma of life, and also biological data banks and databases. Part 4 is sequence alignment, and next time we're going to continue sequence alignment and protein structure analysis, and possibly computational protein design. So here's the tentative schedule for future sessions this year. After today, which is bioinformatics and computational biology, we're also going to have two sessions. So the winners for the third round of 6 sessions will be announced in June of this year. And that's when these 6 sessions end. And bioinformatics is a trial event for Science Olympiad Division C, which is grades 9 to 12 in the 2021 to 2022 school year. So it's okay if you guys don't completely understand everything that's covered today. An introduction of bioinformatics. So bioinformatics is the application of tools of computation and analysis to capture and interpret biological data. And it's very important for the management of data in modern biology and medicine. And the bioinformatics toolbox includes computer software programs like BLAST, which depends on the internet. And bioinformatics analyzes genome sequence data, particularly the analysis of the Human Genome Project. Bioinformatics is a combination of biology, chemistry, statistics, and computer science. And prospects in the field of bioinformatics include contribution to understanding the human genome, leading to discovery of drug targets and therapy. And the Human Genome Project was started in 1990 and concluded in 2003. First, 3 stages of the project: genetic or linkage mapping, physical mapping, and DNA sequencing. Central dogma of life in biology is DNA to RNA to protein. So DNA replicates itself to form 2 strands, and then DNA is transcribed to form RNA, and RNA is translated into protein sequence. And that protein sequence can form amino acids.
How does DNA replicate itself?
We will cover that in future slides. So DNA consists of 4 nucleotides: thymine, cytosine, adenine, and guanine. Adenine pairs with thymine and cytosine pairs with guanine. So Darren, to answer your question, this is DNA replication. So the first step is helicase, which is an enzyme. It unwinds the parental double helix. So it zips the double helix down the middle. And the next step is single-strand binding proteins stabilize the unwound parental DNA. And then the leading strand is synthesized continuously in the 5' to 3' direction by an enzyme called DNA polymerase. And there's another strand that's also being synthesized at the same time. That's called the lagging strand, and this strand is synthesized discontinuously. So there are these gaps that form, and these gaps form segments called Okazaki fragments. And Okazaki fragments can be joined together by an enzyme called DNA ligase. So that's how the lagging strand can then be joined together to form a continuous strand. Now about RNA, there are 4 nucleotides also, but instead of thymine, we have uracil.
Isn't RNA like half of DNA, like 1 strand?
Yeah, it is 1 strand, and DNA is 2-stranded.
Isn't DNA what makes up our genetic?
Yes. So in RNA transcription, what happens is a strand of RNA is synthesized from DNA, and for amino acids are formed from polypeptide chains, and polypeptide chains are basically chains of an amino acid is formed out of 3 nucleotides, each called a codon, and codons are segments of RNA that are transcribed from DNA. So let's say you have a DNA segment, TAG, Then the RNA segment would be AUC. Then you would go onto the codon chart and find AUC. So that's isoleucine. And then that would be the amino acid that you form from the DNA and RNA segment. Okay, so amino acids are sequences are the primary structure of proteins, and there are 20 amino acids that are encoded by the genetic code, and 3 nucleotides can form 1 amino acid. Stages of transcription: there is initiation, elongation, and the RNA polymerase detaching from the DNA. After RNA is synthesized, they're actually modified in something called post-transcription modifications. And in this process, a 5' cap is added to the RNA, and the 5' cap is a modified form of the guanine nucleotide. There's also a poly A tail, which is a sequence of 50 to 250 adenine nucleotides that is present at the 3' end. And the purpose of these modifications after transcription is to facilitate the export of the mRNA and protect the mRNA from degradation by enzymes in the cytoplasm, and also help the ribosomes attach to the mRNA for translation. 2 types of segments within the RNA. This is the introns and exons. So introns are usually spliced out of the pre-mRNA, and exons are joined together. So exons are usually expressed regions. And SNRPs, or small nuclear ribonucleoproteins, they can recognize splice sites or where to cut the mRNA. This is a diagram of translation. So here we have codon recognition. Then it's a peptide bond formation. Then this is translocation. Okay, so for translation, which is mRNA to protein, we have the codon recognition tRNA base pairs with the complementary mRNA codon in the A site, which is part of the ribosome. And the next step is an rRNA molecule catalyzes the formation of a peptide bond, as you can see here. And the third step is translocation, which is when the tRNA in the A site moves to a P site, and the empty tRNA in the P site moves to the E site and is released. Now let's talk about gene density and non-coding DNA. So not all DNA actually codes for the human genome and is expressed. So there's something called non-coding DNA or junk DNA that's actually not junk. So they play important roles in the cell, such as high sequence conservation, but they don't code for proteins, which is why they're not expressed. And pseudogenes are former genes that have accumulated mutations and are now non-functional. And repetitive DNA is present in multiple copies in the genome. And STRs, or short tandem repeats, are a series of repeating units of 2 to 5 nucleotides. And STRs are part of the simple sequence DNA, which is common in centromeres and telomeres. Transposable elements move from one site to another in DNA, and they're present in both prokaryotes and eukaryotes. There are 2 types of transposable elements. One is the transposon. For transposons, they move within a genome by means of a DNA intermediate, and retrotransposons move by means of an RNA intermediate. So as you can see here, this one, the transposon functions on DNA, while retrotransposon uses reverse transcriptase to function on RNA. So in DNA and RNA, there are 2 ends, the 5' end and the 3' end. So the 5' end designates the end of the DNA or RNA strand that has the 5th carbon in the sugar ring of the deoxyribose or ribose. And the 3' end is named because of its hydroxyl group. Sequences are usually written from the 5' end to the 3' end. And if we're given the sequence of one strand, we can easily calculate the other strand. So let's say we have a sequence from 5' to 3', ATCC. Then from 3' to 5', it will be TAGG. Properties of genomes. So most genomes consist of DNA, but viruses actually can be of either DNA or RNA. So that means they can be either double-stranded or single-stranded. So here are a few examples of single-stranded or double-stranded viruses. So in the parvovirus, they use single-stranded DNA. Herpes virus, double-stranded DNA. West Nile virus, single-stranded RNA. And rotavirus, double-stranded RNA.
Can I ask you something?
Yeah.
How can a DNA become a virus? Does that mean we have a lot of viruses in our genetics right now? Yeah, because how is DNA a virus?
Okay, DNA isn't a virus. It should be viruses, however, can be composed of DNA or RNA.
Oh, so DNA can be regular DNA.
Yeah. So there's something called alternative splicing, which happens after the mRNA is modified inside the nucleus. So in alternative splicing, exons can be joined together in different ways. So one segment can be designated as an intron, while another can be designated as an exon. And exons are joined together, so that can produce different sequences. Gene expression. Genes which are being read and transcribed into messenger RNA or mRNA are said to be expressed. So these expressed genes are the ones that are made into proteins. And transcriptome is the DNA transcripts primarily mRNAs being produced at a particular time. For linkage mapping, as you can see here, a linkage map maps a location of many genetic markers on each chromosome, and a genetic marker is a sequence on DNA that can be identified by scientists studying that DNA.
It looks like a barcode.
Does.
Yeah. Recombination frequencies are used to determine the order and distances between genetic markers. So physical mapping usually counts the number of base pairs between DNA. So as you can see here, there's two types, linkage mapping and physical mapping. The three-domain system. So the three domains of the phylogenetic tree of life are bacteria, archaea, and eukarya. Does anyone know which domain humans belong in? So Michelle said eukarya, that's correct.
What? I'm only in 5th grade.
Oh, it's okay. No worries. Yes, the answer is eukarya. Okay. Thank you guys for answering, but I'll give Michelle 1 point for answering first. So many traits found in bacteria first appeared in the ancestors of all the present-day groups, and the split leading to archaea and eukaryotes occurred after bacteria had gone their own way. And the acquisition of eukaryotes by eukaryotes of mitochondria and chloroplasts occurred after their line diverged from archaea. So does anyone know what the process was called when eukaryotes acquired mitochondria and chloroplasts? So what's the theory called that states that eukaryotes acquired mitochondria and chloroplasts to form prokaryotes? No answers? Okay. It's called the Endosymbiotic Theory. So this theory basically states that eukaryotes came from prokaryotes originally, So there were these large prokaryotes that existed, and these prokaryotes somehow ingested smaller prokaryotes that could photosynthesize and perform cellular respiration. And then over time, these smaller prokaryotes just started living inside the bigger prokaryotes, and the bigger prokaryotes eventually evolved into eukaryotes. So that's how eukaryotes were evolved. Okay, so here's our first cohort. What is transcription? Transcription is...
Why is blue and green the exact same?
This one says RNA, this one says DNA.
Oh, okay.
So transcription is DNA being made into RNA. This one would be DNA replication. And this one would be retrotranscription. So that's what happens in some viruses that use retrotranscriptase and synthesis of proteins. That's translation. Where does transcription occur? It happens in the nucleus. What is translation and where does it occur? It happens in the cytoplasm and it's RNA to protein. What is the correct order of protein synthesis? It's DNA to RNA to protein. What are the nitrogenous bases of RNA? This one is AUGC, or adenine uracil guanine cytosine. What enzyme adds bases during transcription? So for this one, DNA polymerase adds bases during DNA replication, and for transcription or DNA to RNA, it's RNA polymerase. What are the three types of RNA? This one is tRNA, mRNA, and rRNA. Three-letter sequence is on mRNA. This one is the codon.
I think nobody picked I don't know on.
Yeah. So for the anticodon, this is the matching sequence on the tRNA, and codon doesn't exist. So it's codon. I just explained this question. So this one should be anticodon. What is a protein factory, or where are proteins made? This one is the ribosome. What are the two main types of mutations? This one is point and chromosomal mutations. So if you have the point mutations, these can be insertions, deletions, substitutions. And for chromosomal mutations, this is like translocations, which is when parts of chromosomes switch and they exchange chromosomal parts. So these are the two main types. What is a protein? A protein is a chain of amino acids or a polypeptide. The role of mRNA as translation is to code for amino acids by matching up their codons with the corresponding amino acid. What happens to mRNA before it leaves the nucleus? So here in this picture you can see these are the introns, and they're being removed here. while the exons are being joined together to form one continuous strand, and a second tail is being added to the 5' end and the 3' end. This involves tRNA and translation to transfer the corresponding amino acid by its anticodon. So now let's talk about biological databanks and databases. So biological data experiences very fast growth. So there are these databases that can store the biological data and information. So here are a few of the few examples of biological data banks. So for nucleic acids or nucleotides, we have the EMBL in Europe, GenBank in the US, and DDBJ in Japan. And for protein databases, there's the PIR, which stands for Protein Information Resource, MIPS, SwissProt, and SwissProt, Trumbull, and NLR3D. For DNA sequence databanks, 3 databanks exchange data on a daily basis, and data can be submitted and accessed at either location. So these are once again the 3 DNA sequence databases. For composite databases, as there are several databases, which one should we search? So composite databases is the answer because they are when several databases are combined, and these make sure that the same search sequence isn't searched twice in different databases. And these are some of the examples of primary databases. For secondary databases, they store information about secondary structure or results of searches of primary databases. So does anyone know what secondary structure in protein is? Okay, so secondary structure in protein is basically alpha helices and beta pleated sheets. So alpha helices usually look like this, and these form from interactions between the nucleotides, and beta pleated sheets can also form, and they look like that. You could do more research on the 4 levels of protein structure after this class if you want. But for primary databases, secondary databases, we have ProCite, Prince, SwissProt, and OWL. Now let's talk about sequence alignment. So anyone can align the sequences so that many corresponding residues match, and residues are basically amino acids or nucleotides. And strong similarity between 2 sequences is a strong argument for their homology. And homology is when 2 or more sequences have a common ancestor. Similarity is when 2 or more sequences are similar by some criterion, and it doesn't refer to any historical process. So homology refers more to history, while similarity is more about the sequence and whether 2 sequences are similar or have similar nucleotides. So to find the relatedness of the proteins or gene, we can use sequence alignment to find the relatedness of the proteins, and mutation in the sequences bring the changes or divergence in the sequence, and they can also reveal the part of the sequence which is crucial for the functioning of a gene or protein. In sequence alignment, there's something called Similarity Search. So Similarity Search searches for similarity, which can indicate conserved function. Does anyone know what conserved function means? This one's a bit difficult. So conserved function? Yeah, sure.
Maybe it's a function that's conserved for a special purpose?
Yes, that's correct. Good job, you got 1 point. So conserved functions are sequences in DNA or RNA that are basically preserved over several species. So let's say if we have a human and a mouse and we compare their genes, if we find a conserved function for, let's say, giving birth, since human and mice are both mammals, that would be a conserved function between human and mice, because that function is the same or that gene that codes for that function is preserved over the two species. So human and mice actually share more than 80% similar genes at the sequence level. And these genes are only a small fraction of the entire genome.
I thought we're also a little bit of part banana, right?
Yes. All species share a small percentage of genes together, even if they're very unrelated. But through evolution, we tend to diverge or have different genomes. So that's completely due to evolution, but some genes are in common between different species. Comparing sequences can help us understand the function of a protein, and locating similar genes in another species can help you understand your new gene. Okay guys, so this part is very important. Scoring a sequence alignment. So here what we have, match score, mismatch score, and gap penalty, and their corresponding scores of 1, 0, and -1. These three form something called a scoring matrix, and a scoring matrix is used to determine the score of an alignment. So here we have an alignment between two DNA nucleotide sequences. So for the matches, we just count how many matches there are. If we count these lines, there are 18 of them. So once we have a matches score, we multiply that by the corresponding match score in the scoring matrix, which is +1. So this would be 18. And for mismatches are any two nucleotides that are in the same position but are different. So that's like A with T and A with C. So these two are different. That's why we count them as mismatches, and we multiply that by 0, which is the mismatch score in the scoring matrix. And we get 0 for gaps. So gaps are basically any spaces in the genome. So here we have A with nothing. So this nothing just basically implies that it's an insertion or deletion. And this entire sequence is shifted to the right because of this gap. So for gaps, we count the number of spaces, 1, 2, 3, 4, 5, 6, 7. So that's 7. And then we apply the gap penalty score of -1. So then we get -7. And once we add all of these scores together, we can get the score of the sequence alignment of +11. I'll give you guys an example if you want to get some extra points. So let's say we have the first sequence is AGCTCG, and the second sequence is C gap CTG, C gap CTCA. So can anyone tell me what's the score of this matrix? I'll give you guys 10 more seconds to answer before I go over it.
What are you asking?
What's the score of this sequence alignment? If we use this scoring matrix here.
I don't know.
So, first of all, we compare A with C. That's not a match. A and C, that's not a match, so we use the mismatch score. So here we have a mismatch of 1. Sorry if it's messy. So far we have one mismatch. Then we compare G with the gap here. So for G with gap, we use the gap penalty of -1. We have one gap. And for C with C, we have a match. So we use the match score of +1. And then we compare T with T, that's another match. So that's another one. Then C with C, that's another match. And finally, G with A. G with A is a mismatch, so we use the mismatch score of 0. So now we multiply all of these and add them together. So for mismatch, we have 2 times 0, that's 0. For gap, we have 1 times -1, which is -1. For match, we have 3. For match, we have 3 times 1, which is 3. So then we add all of these together and we get 2. So good job, Nathan and Ian and I. So I'll give you guys 1 point, actually 2 points each for this question. So there are several ways to create a scoring matrix to evaluate your sequence alignment. So this one is just a point of personal preference. So which one do you guys think is more likely? There's no right answer. Sequence 1 or sequence 2. Okay, so Michelle, Nathan, and Apanav answered. So I'll give you guys 1 point each, but there's actually no right answer. So it basically just depends on what you're looking for. If you're looking for more gaps or insertions and deletions, then you might choose the first one. And if you're looking for a higher score, you might look for a different one. So in order for number 1 to be more likely, we can achieve this by penalizing more for a new gap than for extending an existing gap. So what this means is, number 1, as you can see here, this has many gaps that are consecutive or following each other. So here you can see 7 continuous gaps or existing gaps. So this one might be more likely if we use this scoring scheme here, because compared to the second one, which has many new gaps, we just have an existing gap that's extended. So that's how we can maximize the score if we choose this scoring schema for number 1. For scoring a sequence alignment, we can also use origination and length. So for origination, what this means is the number of total opened gaps that we have. So here we have 2 gaps because that's this one and this one. So that's 2 gaps for the origination. And for length, for the length penalty, we count the individual gaps inside these larger gaps. So here we have 7 of these for length, and then we apply the length penalty, so that's times -1. And when we add all of this together, we get -7. For substitution matrices, they can weigh replacements of one residue by another, and a similar score in a substitution matrix. For similar sequences, we get higher scores in substitution matrices, and these scores are often positive. If we have 2 very different replacements of residues, we have a low score or negative score. And the simplest is the identity matrix. So an identity matrix is when the diagonal going from the top left corner to the bottom right corner is all 1, while the rest is 0. So that's called an identity matrix. Now for PAM matrices, these are derived from alignments of very similar sequences. So PAM1 is mutation is a collection of alignments that are from a mutation event that changed 1% of amino acids. PAM2, PAM3, and so on, they are extrapolated or derived by matrix multiplication. So for example, if we multiply PAM1 by PAM1, we can get PAM2. And to get PAM3, we can multiply PAM2 by PAM1. And there are a few problems with PAM matrices. So these can incorrectly model long-time substitutions since conservative mutations they usually have only one nucleotide change. For blossom matrices, these are derived from alignments of distantly related sequences. And blossom matrices use Black's database, which is an ungapped multiple alignment of protein families at a given identity. And blossom is better for blossom 50 or blossom 62. So you just have to choose the right the right type of sequence alignment based on which two sequences you're comparing. For gap penalties, these can determine the significance of alignment, which depends critically on the gap penalty, and gap penalties can be changed to adjust to the given sequence. So let's say you want to maximize your score for matches, then insertions and deletions or gaps would be less valuable. So if you guys remember what a gap is, that's basically like A to a blank here. So that would be called a gap. And the rules for gap penalties are influenced by knowledge of the structure, and simple rules are used when nothing is known. And there are two types of gap penalties: linear or affine gap penalties. For dynamic programming, it's when optimal alignments are built up using previous solutions for optimal alignments of subsequences. And dynamic programming relies on optimality, which states that in an optimal sequence of decision of choices, each subsequence must also be optimal. And the optimal solution to a problem is when all of the optimal solutions from the subproblems are combined. We'll talk more about dynamic programming more specifically next time. But there are two main types: global alignment and local alignment. So the global alignment was invented by Needleman and Wunsch, and local alignment was invented by Smith and Waterman. And today we have a special. So this is the video that our NASA student launch team from YEA produced, and it's called When Rocketry Meets Biology. Hi everyone, this is our STEM engagement presentation. We're Team NASA Student Launch, Team LALC, and we're from Young Engineers in Action. So a brief overview about our presentation today. First, we're going to cover our team structure, then a brief history of NASA and rockets, then rocket components, rocket building steps, when rocketry meets biology and our team's rocket, then practice questions and a Kahoot, and then it's a Q&A session. So first of all, about our team. We are a team of high school students from California. Our team name is LALC NASA Student Launch Team. Our organization name is Young Engineers in Action. And for our team structure, our captain is Catherine Liu, who is in 12th grade from Oxford Academy. I'm Emily Wren, the Chief Operating Officer. I'm a 10th grader from Diamond Bar High. And our Chief Safety Officer is Lee Marquez, a 9th grader at the California Academy of Mathematics and Science. Our TARC officer is Neil Marquez, who is a 9th grader at the California Academy of Mathematics and Science. Rohan Nair is our STEM engagement officer, an 11th grader at Cypress High School. Andrew Liu is our outreach officer, a 9th grader at Oxford. And Jessica Lee is our finance officer, a 10th grader from Oxford Academy. 1958. And now let's Instagram, Twitter, LinkedIn, and website. Now let's talk about a brief history of NASA and rockets. So NASA stands for the National Aeronautics and Space Administration. It was started on October 1st, 1958 as a part of the US government. NASA is in charge of the US science and technology that has to do with airplanes or space. And the NASA Student Launch Program is a 9-month research and competition program.
Student Launch engages students from middle schools to universities in a 9-month engineering design lifecycle program, mirroring the activities of a spaceflight program. Teams design, build, test, and launch high-powered rockets and scientific payloads. Teams are divided into 2 primary divisions: college, university, or USLI, and middle school, high school, or SLI.
A brief history of rocketry. In 1232, fire arrows were used by the Chinese. In 1591, the first multi-stage rocket was launched by German fireworks maker Johann Schmidlapp. In 1687, Newton's laws of motion were published. In 1844, spin stabilization was invented, and in 1898, space exploration was proposed. 1926, a successful liquid propellant rocket was launched by American Robert H. Goddard. In 1942, V-2 rockets by German engineers led by Wernher von Braun were launched. In 1946, the first atmospheric testing by the US was conducted. In the 1950s, the intercontinental ballistic missiles were fired. And in 1957, the first satellite, Sputnik 1, was launched. Now let's talk about the components of a rocket.
So the basic outside structure for our TARC rockets include the nose cone, upper body tube, lower body tube, fins, and then the motor containment or motor mount. And the inside structure is in the upper tube, we have an egg and altimeter. This year it's 2 eggs, and the under tube is going to have a parachute and a rocket motor, which is secured by the motor mount. The outside components are made of these body tubes. These ones are made of cardboard, and they hold the inner parts together during the launch. It's important to cut the tubes to the correct length so the rockets can be stable as tested on open rocket software or other softwares. The nose cone is to reduce the drag on the rocket, and we use parabolic, I think, and there's parabolic, ogive, blunt, and hemispherical. There's also other nose cones that people have experimented with, and we use a plastic 3D printed nose cone. The purpose of the transition piece is to help the 2 tubes of the rocket transition and separate during launch. And we use cardboard for our transition pieces. The bulkheads are connected to the transition pieces and also connected to the parachute. So when the separation occurs, there is a parachute to help prevent the rocket from falling too fast. The fins are to maintain rocket stability. Without them, the rocket would just like spin out of control. So you need to use them. And there are two fins which we use on our Tark rocket. There's also these other fins, delta fins, which are these ones are made of wood. And we have a motor mount. This is important because it holds the motor and secures it so the rocket can fly. And if the motor falls out, it's very dangerous. So you don't want that to happen. Now, a recovery system is one parachute, and the importance of a recovery system is so you can reuse the rocket and recover it after it's landed. Otherwise, it would just explode on impact. And on larger rockets, there may be tracking systems because they may glide farther than expected. Our payload has 2 eggs and an altimeter so we can record flight data. Otherwise, we would not know how high the rocket went. Now, these are the basic rocket building steps. These are the basic rocket. Okay, so steps are to cut both body tubes to desired lengths, cut tube fins to their desired lengths, glue the transition pieces and the bulkhead, 3D design the motor mount, and that can be attached, add in rail buttons or launch guides, secure the tube fins to the bottom tube, drill holes for the nose cone and altimeter, make a case for the egg so it'll stay safe, add a recovery system, parachutes, and spray paint the rocket for extra thickness. For the first step, to cut both body tubes to desired lengths, we use hand saws or we can use knives or X-Acto blades. And we use a box that has a V-shaped cut into it to hold the tubes in place while we're cutting them. For the second step, cut tube fins. We usually use 3, but 6 as pictured in this rocket, we also use that. And the most important thing is that the fins need to be spaced evenly. And for the 6-finned rocket, we don't need to think about that because it'll just fit perfectly. And delta fins will be can be also used, like we use delta fins on our NASA student launch rocket. So for step 3, we glue the transition piece and the bulkhead. So we pop out the bulkhead centering rings and from the plywood that is laser cutted, we get the screw eye and twist it into the bulkhead. And when we put it mostly all the way in, we will glue it with epoxy, and this will secure it permanently to the bulkhead. And we will epoxy the bulkhead to the smaller tube, the top tube, and we will also glue the centering rings on the outside of that tube. And we will wait for 1 day for the epoxy to dry. For the motor mount, we use 3D CAD to design the motor mounts. This is one that is not 3D printed. It's wood with the centering ring inside, but the motor mount ring inside. And this is hard to build, so we just 3D print them. For step 5, we drill holes on the bottom tube and we put motor rail buttons or launch guides into the holes. For step 6, we secure the tube fins. We mark holes on the tube fins and the bottom tube, and we're going to drill holes in the bottom tube and the two fins, and we screw them in and we epoxy the fins to keep them in permanently. For step 7, we drill holes for the nose cone and for the altimeter. So we have a 3D printed nose cone. So there are 2 holes on each side. So we just drill holes, make sure they're aligned with the nose cone, and we also drill holes in the top tube so that the altimeter can read the pressure correctly. For step 8, we need to make a case for the egg. We use foam from a memory foam pillow, or we use foam from pool noodles, and both these are good options. We take measurement of the egg and we cut the foam into a shape of an egg so that the egg can be inside the foam, and we test if it's good or not. Step 8, we add a recovery system. We use Kevlar rope, blast protector, and parachutes. The blast protector is so that during the blast, the blast won't burn a hole through the parachute or burn the rope of the parachute. This way, the rocket can come down safely every time. And we have 2 parachutes, one for the bottom, one attached to the bottom and one attached to the top. And we tie rope around the motor mount, and we tie the blast protector away from the parachute, and then we tie another rope to the end of the bulkhead, and another parachute. For step 10, the last step, we spray paint the rocket, and we usually just spray paint it one solid color, but designs can be added if you want.
When Rocketry meets biology, our team's rocket. So here's an overview of the dimensions for our NASA student launch rocket. The length is 183 centimeters, weight is approximately 16 pounds. Apogee, or the highest height that it flies, is 4,501 feet. 1 feet. The maximum velocity is 525 feet per second, and maximum acceleration is 241.5 feet per second squared. And our thrust to weight ratio is 5.7 to 1. The length distribution, the nose cone is 58.4 centimeters, body section 1, 40.6 centimeters, body section 2, 83.8 centimeters, and the total drift of the rocket with 20 miles per hour wind is approximately 2,091 feet. The materials we used for our rocket, the main body, nose cone, fins were all made out of fiberglass. The streamer and parachute were ripstop nylon, and the glue was epoxy glue. And we decided on fiberglass because it's highly durable and resistant to most forms of moisture and corrosion. And it's also a good compromise between weight and strength. And we used ripstop nylon for the streamer and parachute because it's durable and doesn't break easily. And we didn't consider other options due to convenience of purchasing. For our rocket recovery system, at apogee, the streamer is deployed from the bottom compartment, and it will be 6 inches by 30 feet. And at 600 feet, the 72-inch diameter parachute will be deployed from the first compartment. And both recovery systems use black powder ejection, which is connected to an altimeter in the avionics compartment in between the 2 rocket body tubes. And for our subscale flight results, we had a successful flight to 2,193 feet with successful deployment of the streamer at apogee and parachute at 600 feet. We also performed a successful ground test prior to launch. And we also changed the location of one black powder for the streamer to come out more easily. For our payload, the purpose is to conduct an experiment on how forces and altitude affect mimosa pudica plants. The tumbler was designed to prevent plants from turning upside down, and the 3D printed capsule is replicable. And for our mimosa pudica plants, there are 3 inside the rocket as our experimental group and 3 on the ground level as the control group. And we're going to have 2 cameras inside the tumbler in order to document the process of the opening and closing behavior of the plant's leaves. And we installed lights inside the tumbler to ensure that each plant receives the same amount of light. For our tumbler design, it's designed so that the plants can always stay upright, and our tumbler is sphere-shaped, and a weight is to be placed at the bottom, and the tumbler is easily removable from the rocket tube after the launch. For our mimosa pudica plants, we conducted extensive research on the biological properties of mimosa plants and previous experiments conducted on them. So by preliminary research, our team found out that the earliest related experiment was conducted by a scientist named Prefer in 1873, and that experiment showed that repeated mechanical stimulation of mimosa leaflets led to a decrease in its sensitivity. And 2 of the recent related experiments used the leaflet closure as a dependent variable and explored the vertical dropping light intensity and other factors as the independent variables. Some other recent research on mimosa plants focused on the cellular level instead of the behavioral level of biological properties and the learning behavior of mimosa plants. But based on our team's preliminary research, it seems that none of the previous experiments about plant cognition on mimosa plants were conducted in flying rockets, and all of the previous experiments were conducted in a lab on the ground level. And our team is curious to see how different forces and altitude, which are the independent variables, would affect the leaflet closure behavior, which is the dependent variable in flying rockets. And our team thought it would be interesting to explore how rocketry, space engineering, biology, psychology, and cognitive science would interact with each other in our team's experiment. And to wrap it up, today we covered an introduction of bioinformatics, the central dogma of life, biological data banks and databases, and sequence alignment. And next time we'll be continuing with sequence alignment, protein structure analysis, and computational protein design. So our next session is going to be on March 21st, and we'll also be covering bioinformatics and computational biology. It's going to be at the same time, 5:30. So I'll see you guys again. Bye.
Bye.
People who kept your cameras on the entire time, I'll give you guys 5 extra credit points.
Woohoo! Bye.
Bye.
Goodbye.
I can turn off my camera now, right?
Yeah.
Goodbye.
Bye.
Thank you.